My name is Joshua Mugabi. I welcome you to Topic 1 Cell Biology. Our learning outcome D. Analyze the structures of plant. Parenchyma, parenchyma, sclerenchyma, xylem and phloem. And animal, epithelial, cardiac, aeraria, fibrous and skeletal tissues to assess their roles in physiological processes, disease diagnosis, and levels of organization. Our learning activity is B. In pairs, learners conduct an investigation to compare micrographs of healthy tissues from the internal organs, the liver, the lungs, the heart, or intestines of a mammal with images of micrographs of diseased tissues from the same organs. They make a report and represent the findings to the class. Comparison of healthy and diseased mammalian tissue. Item 1. We are going to look at comparative histology investigation. Students are provided with labeled micrographs of healthy and diseased tissues from the liver, lungs, heart, and intestine of a mammal. They are required to observe, compare, and explain structure differences. Task. State the aim of the investigation. Identify 2 organs studied. List 3 observable differences between healthy and diseased tissues. Suggest possible causes of the observed changes. State the aim of the investigation. To compare the microstructure of healthy and diseased mammalian tissues. Identify 2 organs studied. The liver and the lungs. List 3 observable differences between healthy and diseased tissues. Loss of normal tissue arrangement, presence of damaged or dead cells, thickened or inflamed tissues, reduced air spaces, that is in the lungs, accumulation of fat in the liver. Suggest possible causes of the observed changes. Infection by pathogens, poor nutrition, toxins or chemical exposure, chronic disease conditions. Comparison of healthy and diseased mammalian tissue. Item 2. We are looking at the lung tissue comparison. Students observe micrographs of healthy lung tissue and lung tissue affected by pneumonia. Task: describe the structure of healthy lung tissue. Describe changes observed in diseased lung tissue. Relate these changes to lung function. Describe the structure of healthy lung tissue. Thin-walled alveoli, large air spaces, rich capillary network. Describe changes observed in diseased lung tissue. Thickened alveolar walls, presence of fluid or pus, reduced air spaces. Relate these changes to lung function. Reduced surface area for gaseous exchange, slower diffusion of oxygen and carbon dioxide. Comparison of healthy and diseased mammalian tissue, item 3. We are going to look at the liver tissue comparison. Micrographs of healthy liver tissue and liver tissue affected by fatty liver disease are provided. Task: state 2 features of healthy liver tissue. State 2 features of diseased liver tissue. Explain how the disease affects liver function. State 2 features of healthy liver tissue. Well-organized hepatocytes and clear sinusoids. State 2 features of diseased liver tissue. Fat droplets within liver cells. Distorted tissue structure. Explain how the disease affects liver function. Reduced metabolic efficiency, impaired detoxification and bile production. Comparison of healthy and diseased mammalian tissue, item 4. Intestinal tissue comparison. Students examine micrographs of healthy intestine and intestine infected with parasites. Task: describe the structure of a healthy intestinal tissue. Describe structure changes in a diseased tissue. Relate the changes to nutrient absorption. Describe the structure of healthy intestinal tissue. Numerous villi, thin epithelial lining. Describe structure changes in diseased tissue. Damaged or shortened villi, inflammation of the intestinal wall. Relate the changes to nutrient absorption. Reduced surface area for absorption and poor nutrient uptake. Comparison of healthy and diseased mammalian tissue. Item 5. Heart tissue comparison. Students observe micrographs of normal cardiac muscle and diseased cardiac muscle. Task. Identify two features of healthy cardiac muscle. Identify two changes seen in the diseased cardiac muscle. State one effect on heart function. Identify two features of healthy cardiac muscle. Regular striations, intercalated discs. Identify two changes seen in diseased cardiac muscle. Damaged muscle fibers, scar tissue formation. State one effect on heart function. Reduced force of contraction, poor blood circulation. Marking emphasis. State clear aims. Make accurate observations. Compare healthy and diseased tissues. Relate structure to function. Draw varied biological conclusions. Conclusion. Disease alters the normal microstructure of tissues, leading to reduced efficiency of organ function. Thank you for participating in Topic 1, Cell Biology, our learning outcome D. Analyze the structures of plant, parenchyma, collenchyma, scalenchyma, xylem and phloem, and animal, epithelial, cardiac, areolar, fibrous, and skeletal. Tissues to assess their roles in physiological processes, disease diagnosis, and the level of organization. We have been looking at learning activity B. In pair, learners conduct an investigation to compare micrographs of healthy tissues from internal organs, liver, lungs, heart, or intestines of a mammal with images of micrographs of diseased tissues from the same organs. Make a report and present the findings to the class. If it has been useful to you, please visit me at Mugabi Joshua Biology on YouTube.